uh, look to science to tell us how to understand the world around us, the universe around us, as well as how to understand ourselves. So that's why a science lecture fits pretty well within the idea of humanism. So the title of the talk is The Neuroscience of Memory, Return of Engrams. So the talk is largely based on these two review articles, one from Science and one from the Annual Review of Neuroscience. They have the same first author, Sheena Jocelyn, but I've also looked at some of the underlying original publications which were quoted in these review articles to get a better, more detailed understanding of the experiments, which I will attempt to explain to you. Okay, why do I say return of the engrams? Well, that's because the idea is really very old. It was coined in 1905 by this fellow named Richard Simon, and he quoted he coined the term because he was thinking about how does memory form and can we recall a memory? And this was very early days, of course. So he thought, well, there must be something happening in the brain, some change, either morphological or biochemical or something. There must be some change where the brain can store the memory. And upon recall, that part of the brain is activated once again. And he defined an engram as the enduring, though primarily latent modification in the irritable substance produced by a stimulus. So that was one of the first ideas of how we can actually store memory and then retrieve the memory. Now, Simmons was pretty wise, and he did not try to understand the mechanism by which engrams worked, because he realized that the science was just not there yet. And for several decades, scientists attempted to prove that engrams existed in mice, and no one was very successful, so the idea sort of fell by the wayside. And that's why we're talking about the return of the engrams, because in more recent years, with new technologies being developed, it looks like the engrams are really a valid concept. And Donald Hebb is another scientist who published I guess it was a book, The Organization of Behavior, in 1949. And he tried to postulate mechanisms by which memories were stored and recalled. So this is very much along the line of the engram, but he came up with a idea of actually how it works. He defined, I'm not sure he actually used the word engram, but he probably used the word memory trace, but essentially they're talking about the same thing. He said, memories are formed when cell assemblies form strong interconnections at the time of memory formation. These neurons are reactivated upon memory retrieval. And this is succinctly stated very famously as neurons that fire together, wire together. So basically, this is when you combine what Simon and Hebbs were both saying, this is the idea behind an engram. It's a group of reciprocally interconnected neurons that are simultaneously active during an event. The neurons are reactivated upon retrieval of the memory. Growth or metabolic changes that strengthens the interconnection between assembly cells result in an engram. Engrams are not strictly localized to one particular cell or brain region, but may be distributed among many different structures in the brain. Okay, so obviously much of this work that I'll be talking about, if not virtually all of it, as a matter of fact, will be work that's done in mice. So the obvious reason is a lot of these techniques that we use are invasive and can't be used in humans. So we'll be looking at memory. So how do we know what a mouse remembers? We can't ask it, but there's been a lot of experimental procedures that have been developed to inquire what a mouse remembers. One of the most widely used, and the one that I'll be talking about mostly today, is context fear conditioning. And in this process, you have mice, say in the blue cage, which is their home cage, and they're presumably happy there. They eat, they drink, they do all that mice do. Periodically, they would be put in a different colored cage or a different cage. I'm using blue and red just as a shorthand. So you put it in the red cage and subject it to a mild shock, and the mouse doesn't like that. So eventually the mouse will associate the red box with being shocked. And when the mouse is shocked, it will freeze, meaning it just doesn't move at all. This is a normal response of mice in nature. If a cat's sneaking up on it, it may run if it thinks it can get away, but it might just freeze, hoping the cat won't see it. So it's a normal response that mice will do when they are in a situation where they may experience fear, although we realize that we don't really know what a mouse is experiencing subjectively, but the behavior makes us think it is a fearful behavior. So then after the mice have gone through this kind of training, they are put in a red box again, but this time there's no shock administered. But the mouse remembers being shocked in the red cage, and therefore it will remember the shock, and it will freeze anyway. So this is a good way of measuring the memory of the mouse, that it remembers being shocked in the red cage. And of course, the lower part of the slide shows the control mice, which were not shocked, and therefore they don't freeze when they're placed in the red cage. So you can see on the right, the graph, this is actually some real data. The NS means non-shocked. So these animals, actually about 20% of them do freeze. So it's not as if none of them freeze, but 20% of them do freeze. The orange bar are those that do go through the association of the red box and shocking. And there we see, it looks like around 60% of the mice do freeze. So it's not all or nothing. It's not 0 and 100%, but this is the kind of data that you get. It's biology, so the data is not always as clear cut as we like, although that's a pretty hefty, robust difference in the two situations. Okay. All right. So we talked about the engram being composed of neurons, and we know there are billions of neurons in the brain. And these little stars here, this is my little effort to represent a neuron. So pretend these are neurons. And we know that engrams are distributed among many different brain regions. The two that we'll be mostly interested in are the hippocampus and the amygdala. Hippocampus particularly because it is involved in most memory formations, at least episodic memories, which are the kinds that we'll be talking about today. And the hippocampus also enables an animal to, for example, if there's a maze, to learn how to navigate a maze. So it's very much involved in an animal knowing about its surroundings. So it'll be very important in the experiments that we'll be talking about today. The amygdala is an area that we often say is associated with emotions, but in reality, we have to say it's a region that is involved with valence, whether something is the mouse likes or something the mouse doesn't like. The amygdala will be very much involved in that, and we'll see a little bit more of that later. So the mouse brain, many different areas can be involved with forming the engram. We've already talked about the hippocampus and the amygdala. If there's a memory of an odor, then the piriform cortex will be involved. If there's an aversive taste, the insular cortex, spatial memory, retrosplenial cortex. And when we start talking about long-term memory, which we will talk about a little later, the medial prefrontal cortex will be involved. So clearly many different regions and structures in the brain can be involved in memory and engram formation. Now, neurons are not static. They undergo changes, and at some stages of this change, they're referred to as being excitable. And that simply means that they can be easily triggered to fire. So another cell, another neuron can give it a weak signal and it will fire. Well, those that aren't excitable, which are the black ones here, they cannot easily be fired, so triggered to fire. So the blue ones are those that are excitable. And engrams will preferentially incorporate the excitable neurons into the engram and exclude the engram, the neurons which are not excitable. That's an important feature of memory formation in the mice. Okay, so this is a great hypothesis. Does it really exist? So these are 4 questions that I actually took from the review articles. And so we're going to go through experiments that relate to these various questions to come to some belief that engrams either do or do not exist. And the first question is, can memories be turned off and on artificially? Can we develop a way of doing that? And I'm going to show you a short video on optogenetics because this is extremely important technology for several of the, in particular, the next one experiment that I'm going to show you. So this is a fairly recent technology, and the video is pretty short, but it'll give you an idea of what this technology is all about.
This is optogenetics. It all started with algae. In 2002, researchers discovered that a protein that causes green algae to swim towards or away from light is a light-sensitive channel. Blue light causes the channel to open, and positive ions then flood into the cell, much like the influx of ions that cause nerve cells to fire. Neuroscientists were quick to see the potential of these algal proteins. They realized that if they could get proteins called channel rhodopsins to work in mammalian nerve cells, they'd have a very precise way of controlling nerve activity in the brain. For many applications, this method would be better than drugs, which act too slowly, and better than electrical stimulation of the brain, which is less precise. Getting an algal protein into, for example, a mouse brain requires a bit of genetic engineering. First, you take the gene that encodes the protein, plus another piece of DNA called a promoter, and you put them in a virus. When the virus is injected into the mouse's brain, it infects neurons and delivers the gene. A chosen subset of those neurons will have the right machinery to activate the promoter, and in those cells, the algal protein is expressed in the cell membrane. To activate the protein, all you need is light, delivered via a fiber optic cable. It's the ability to target specific neurons in living, moving animals that makes this technique so powerful.
Okay, so that was about optogenetics. Just another word about the gene that they talked about. You express the gene, which is sensitive to light, which will allow us to fire the neurons at will. A word about the promoter. This promoter is designed so that it is only active in a neuron which has been activated. So that's how you get specificity. The virus may infect a lot of cells, but only those cells which, neurons which are activated will actually make the protein. And by the way, the virus is only a vehicle, so it's not really a replicating virus. It's just a vehicle for getting the gene into the right cells. Okay, so let's look at this procedure again, where mice are shocked and they freeze as a response, and they form engrams. And you can see here the neurons which are blue are the ones that were active, and therefore they get incorporated into the engram and form strong bonds to one another. And that's indicated by the line there. And you can see that both cells in the hippocampus as well as the amygdala are incorporated into this engram. So we move the animal now back to its blue box, and of course it's not afraid of being shocked because it doesn't have a history of being shocked there. So the engram is still there, but it's silent because I've made it black now. But we have genetically engineered that engram to contain the light-sensitive protein. So we can use this to turn the neuron on and off. So once we shine the light into the mouse's brain, then the mouse thinks of being shocked, although it's never been shocked in a blue box, but it can remember being shocked because we've turned that engram on, that memory of being shocked. And that will cause that engram to fire, and it will cause the mouse to freeze. So here we've turned on the memory using artificial means. So the data at the lower left shows you the percentage of the mice that freeze under this condition. So if the light is off, virtually none of them freeze. If you turn it on, it looks like about 25% of them do. A fairly low number, but still a very significant number. And you can see how you can turn the memory of being shocked on and off. Okay, so can memories be turned on and off? And I would say that that data says yes, it can. I would say yes, in that the amygdala is known to respond to pleasant stimuli also. Unfortunately, most experimentation are dealing with the negative, however. You know, I guess the negative is a stronger incentive for the mouse to respond, and probably that's why. But I would say yes. I would think the memories would be positive memories would be held in the same, would be formed in the same way. Yeah, I would be more hard pressed to come up with data to prove that, though. It's a good question. What the mouse is experiencing subjectively, I'm not sure, but the memory is of a shock, and so that memory is turned on. What the mouse is subjectively experiencing, does he think he's in a red box or what? I'm not sure. Yeah, so the engram is composed of neurons, and as you might remember, neurons have axons, which are these long extensions on one end of them, as well as dendrites on the other end, and that's where the signal goes from one cell to the other cell. And actually, those axons can be quite long. You know, we have nerve signals have to get to very remote parts of the body, so just moving around the brain is really not much of a problem for a neuron. Okay, so we'll go to the second question. Are neurons active at time of memory formation, also active when memory is recalled? And so that would be the idea of an engram that it's formed, and then when you do a recall, the same neurons would be activated, right? So let's see if that's true or not. Okay, so here we go. We shock the mouse, he freezes. Let me see, I think is that the right slide? Oh yeah, okay. So using techniques pretty much like the technique that we saw with the optogene, scientists can specifically engineer or change neurons. And in this case, rather than putting an optogene in, we put in a fluorescent protein, which fluoresces red. And so I've drawn the engram in red here. You put the animal in the blue cage, it's no longer remembering the shock in the red cage, so the engram becomes silent. It's still there. The cells are still fluorescing red, but the engram is silent, and I've shown that with a dotted line. Now we put the animal back in a red cage, and it remembers being shocked, of course, so it activates the engram. And now we label the cells which constitute the engram with a green label. So the cells that were there when the memory was formed are red, and those that were there when the recall was made are green. So the question is, are these the same cells? So this is looking at the amygdala of a mouse that has undergone these kinds of manipulations. And we see during memory formation, there are a few cells here that are red. So memory formation cells, they were active, are red. And then at the memory retrieval, we can stain the cells green. And then we can superimpose these two panels, and we see we still got some red cells, and we still got some green cells, the green cells. The green cells are hard to see because you're actually staining the nucleus, and it's much smaller than the entire cell. And, but you do see some that seem to be both green and red. So those are the cells which were active at the time of the memory formation, as well as at the time of memory retrieval. So this would support the idea that the engram is a reactivation of neurons which were active at the time of memory formation. And you see at the lower right-hand left, you see actual data. The orange is the percent of cells which are both red and green, and the gray bar shows the in the controls the number of cells, a percentage of the cells which are red and green. Again, it's not an all or nothing thing, but the number in the fear conditioned animals is much higher than in the control. Okay, so our memory is active at time of memory formation, also active when memory is recalled. So answer, I think, is yes. All right, so if we believe that memories are encoded in engrams, if we destroy an engram, can we destroy a memory? Very specifically destroy the engram. So here, again, use the same techniques that we've talked about earlier with putting the virus into the mouse brain. And now you are expressing a diphtheria toxin receptor in the engram cells. Now, mice are not susceptible to diphtheria toxin because their cells don't have this receptor on them. However, we've very specifically labeled the engram, so now it does have the diphtheria toxin, so it should be susceptible to the toxin. So when we treat the mouse with the toxin, that engram, and only that engram, will be killed. And when that happens, the mouse doesn't freeze anymore. It doesn't associate the red box with being shocked. Okay, this is some of the real data. The green box are the percentage of animals that freeze before we add the toxin, and the black box is the percent of animals that freeze in the absence of the toxin. So if the engram is destroyed, is the memory destroyed? I think the answer would be yes. Okay, the final question, will insertion of engrams for an event that never happened result in a false memory? So can we implant a false memory into the mouse's brain? Well, here we are, the mouse is in its home cage, but it forms an engram for the home cage also. It's just not linked to the amygdala because there's not been a shock. But we've actually labeled these cells again with the optogene, so we should be able to turn these cells on and off. So when we put an animal in another cage, say a green cage, one that it's never seen before, the blue cage engram will become silent because it's not in the blue cage. Now, if we turn the light on, we've turned this engram on, this one here, and the mouse is thinking blue cage. So if we shock the animal now, then it forms the engram linked up with the amygdala. So it's now associating a blue box with being shocked, even though it was never shocked in a blue cage. And so it freezes. So is this, I think we have shown that we have inserted a false memory into the mouse. Okay, so false memories are problems not only with mice when they've been engineered, but false memories are a problem with humans also. And this is very specifically true in the case of criminal law. There's been, at the time of this article, which was written in Scientific American in 2017, there were 349 wrongful convictions that were overturned based on DNA evidence. And 70% of those, the reason the person was convicted, the primary reason, was through eyewitness testimony. And the paper points out very interestingly that if you have like blood as a part of evidence, then there is a chain of custody, so that every stage they have to know who has had access to the blood and what they did with the blood. And that has to be very well documented for it to stand up in a court of law. But with memory, that's not the case. There is no chain of custody, and therefore the memory can be contaminated. So the article suggests that we use only initial memory, or at least we know what the initial memory is in case it changes later if the witness changes, then we have to ask why. Has the memory been contaminated in some way? And certainly the certainty, if the person initially is not very certain that a particular person committed a crime, that needs to be recorded also, because they may become more certain later if their memory gets contaminated. Now, how can one's memory get contaminated? Well, we don't really know that humans have engrams, but we know if we use the model of the engrams, however, we could come up with a hypothesis that the neurons become excitable when a memory is recalled, right? And they remain excitable for a while. So if a new engram is formed by a new memory, then they may become linked. And therefore the eyewitness will not realize that their memory has been corrupted, and through no fault of their own, they may come up with a false memory. So that's why we have to be very careful with eyewitness accounts in courts of law. Okay, let's see, how are we doing on time? Okay, so I have a lot of things in the chat box. I haven't had a chance to look at them, but I thought we'll stop sharing right now and we can entertain a few questions. Anyone have a question? Yeah, memory retrieval is another area of research, which I'm not going to go into right now, but certainly when people age, for example, it's sometimes hard for us to retrieve the memories as well as it is as well as we can when we're younger. We will go into toward the end of the talk, into long-term memory versus short-term memory and how those differ, so that might have some relevance to your question when we get to that. Very good question. We will talk about the extinction of memories in mice, and we'll also talk a little bit about how PTSD, for example, efforts to treat PTSD and people trying to erase memories, or at least association of bad memories with certain stimuli. So I guess I don't really know the answer, but we will touch on some of those issues. Okay, so memories do not exist in isolation. And this is a quotation. Frankly, I can't remember where I got the quotation, but it's a good one anyway. The simultaneous retrieval of multiple engrams with similar content could provide a basis for complex cognitive processes such as generalization, abstraction, and knowledge formation. So memories get linked together, they get associated with one another. And this presumably allows us to do higher thinking. And so we'll talk a little bit about how memories get linked together. And we've talked about excitability and how for a neuron to get incorporated into a memory engram, it has to be excitable. And we know that when a memory is retrieved, the neurons are excitable. So it's possible when you're thinking of something that is you have a high probability of a new experience, new information getting incorporated into that same engram, or at least linked to that same engram. And that's how we associate things together. And we all have had strings of thought, how one thought leads to another thought, which leads to another thought. So this may be the mechanism by which that happens. So we'll look at some experiments that address that. Okay, so this is a little bit of a different protocol, but it's sort of the same thing where there's a tone, a noise, or sound. When a mouse hears it, it's shocked and associates being shocked with that tone. And it'll, of course, form an engram. So let's say the blue engram for tone 1. If you do the same thing with a different tone, you will form a new engram, which is shown in red. And if you do this within 6 hours, then the two engrams overlap a lot. They share the same neurons. So we can see how we could easily trigger both engrams. However, if we wait 24 hours and do the same thing, the two engrams are very distinct. They don't share many neurons, so they're not linked together. Okay, so now we talked a bit about memory extinction, fear extinction. So this is how scientists look at that. Again, they do the training, the association of shock with the red box. Then they put the mouse in a red box, but they don't shock it, and they do this several times, many times. And eventually the mouse stops freezing in the shock. It has forgotten the relationship between the red box and being shocked. So its fear memory has been extinguished. Now, how does that happen? There's two possibilities that I can think of. One will be that the engram for that memory simply decays, lack of use, use it or lose it. The other possibility is the mouse has undergone new learning, that it has learned that the red box is not necessarily associated with shocking. And if it's learning something, a new engram must be formed. So in fact, it's the latter. A new engram is formed. So the old engram is still there. This is a diagram of the amygdala of a mouse. And there are actually different cell types there. We talked about, because we're usually looking at negative events, how about positive events? Will that create engrams also? And I think surely it does. Certain cells in the amygdala will respond to negative events such as shock, and therefore they will be incorporated into an engram. Other parts of the amygdala respond to positive events, like not being shocked in a red cage. And so therefore the extinction engram will probably be formed in that part. And these two engrams probably interact, and one suppresses the other depending on which one is stronger. And so this is how a memory can be extinguished in a mouse. So this may have relevance to things like post-traumatic stress disorder and things like anxiety disorders. So I'm not an expert in these areas at all, but I did a quick survey or a quick check on Google to see how people try to treat those who are suffering from post-traumatic stress disorder. One that I looked at looked very interesting: eye movement desensitization and reprocessing. Well, the eye movement part doesn't make sense. That's the patient will move his eyes following the therapist finger moving back and forth. I don't know what that does, but I thought this other sentence was very interesting. The goal is to be able to think about something positive while you remember your trauma. It takes about 3 months of weekly sessions. So perhaps what they're trying to do is to extinguish the association of the traumatic, the fear that's induced with loud noise. For example, if someone's in a war, a bomb goes off, there's carnage of human beings, and so loud noises become associated with this horrible situation. But if this can be changed so that the loud noise, a firecracker or whatever, doesn't set off this negative experience, then perhaps post-traumatic stress disorder can be treated. Now, I don't know how successful this is. My impression is there's nothing that's really effective all the time. And so this is just one that I was able to find by Googling. Okay, now let's talk about long-term and short-term memory. I think someone talked a little bit about that. So certainly we know that memories formed recently are different than memories formed in the distant past. And we know this because people suffering from certain forms of amnesia and dementia will forget recent memories long before they forget more distant memories. So they're different. So what's happening here? Well, this is an area of ongoing research, so I'm not sure that all the answers are here, but something does happen when memories age. So this is a recent memory, and as before, we talked the hippocampus plays a very critical and central role in this. And if this is putting an animal in a red box and shocking it, then the amygdala will be involved, and hippocampus will have strong interactions with the amygdala and maybe other areas. This happens to have endorhinal cortex. And also the hippocampus will have strong connections with the medial prefrontal cortex. Now the strange thing is you can put the mouse in a red box and you can see the engram and the hippocampus, the amygdala, and the other parts of the brain, but you can't see it right away in the medial prefrontal cortex. And you know it's there because you can do the optogenetic trick and turn it on artificially. So, but if you wait for a time, for a few weeks later, now when you put the animal in the red box, you can see the engram in the medial prefrontal cortex, and it actually is playing a very important role in its interaction with the rest of the brain. Hippocampus, its role may be changing, diminished, but I think this is still an area of experiment. A lot of experiments are ongoing. All right, so virtually all the experiments I've told you about so far have been in mice. And of course, we were interested in mice, but we were mostly interested in what it means about the human brain. And of course, the reason all this is done in mice is because these are pretty invasive techniques and we can't do them in humans. However, there are some very exciting technologies that have been developed over the last several years that we can look into the human brain without an invasive technique. And this is functional MRI. And you can scan a person's brain and you can scan them while they're doing things, or you can ask them to think about something and scan it and see what the brain is doing. Now, the only downside of it is it doesn't have high resolution. So you can't look at individual neurons or individual engrams. You can only look at regions of the brain. And so here's an experiment. I can't say I fully understand this experiment, but I'm showing it to you because the authors of this experiment, which is in humans using functional MRI, said that the reason they were doing it was because of the data in mice. And so I think the data in mice will inform scientists who are working in humans. And this is one of the advantages of doing experiments in animals. But basically, as I understand it anyway, on the x-axis, this shows you the overlap, the degree of overlap of the functional MRI signal when memory is formed for a particular word versus when it is remembered. And the y-axis is a measure of whether the words were remembered in the correct sequence. So their conclusion was that that something whether it be an engram or something similar to an engram is being linked in the human brain. So for what it's worth, it is at least people are trying to see to what degree experiments in animals are relevant to humans. Okay, with that, we are finished with the presentation. So any discussion, questions anyone would like to bring up? Yeah, since I was focusing on the basic biology of engrams, of course, this is essentially all done in mice, which don't have language as we understand it anyway. There are many different kinds of memory. This memory we're talking about is episodic memory, remembering of an episode that happened to you or to the individual. Semantic memory is memory of facts, which are expressed in language. I think is a different beast. I don't know exactly how these interact. I think that's very interesting. There is the idea that your memories get interlinked and they form a schema, which may be a philosophical or a worldview. And once that is formed, because we all know that there's cognitive biases and people look for information that supports what they believe already. Those biases, I think, are hard to break. I think you can because people do change, but exactly what's going on in the brain, I think, is tough to know. But people are looking with dysfunctional MRI, so maybe they will come up with more definitive answers to that. I think that's the experiment in the when you do the extinction of fear in the mice, that they simply form a new engram to counteract the first one. But the first one's still there. If you put that animal back in a red box and shock it, I'm sure it'll very quickly relearn to fear a red cage or red box. Yeah, both are still there. The idea is that one engram will, with time, hopefully, and experience, will be able to suppress the other one. So they suppress one another, and one, depending on experience, one will be stronger than the other. It's my understanding of the experiment. It's consistent with the engram hypothesis, in that whenever you recall something, you have activated or made your neurons in that memory excitable. So it's easy for new information to either get linked into that engram or actually incorporated into it, which is where false memories come from, we think. Yeah, I think you're right that you're actually remembering the last time you remembered and what happened to it. You know, how did it get changed? And then the next time you remember the last memory. So I think what you're saying is absolutely true. With Alzheimer's, for example, eventually the brain is, much of it is destroyed. So they're just, you know, the engrams are destroyed, I guess. That's probably advanced forms, but there are intermediate stages where they just can't recall things, but the engrams are probably still there. That's just the recall, whatever process that is diminished. And you know, we talked about in some cases in the mouse, that for example, we couldn't see the engram in the prefrontal cortex when there was a new memory, but we could see it later on, and it was there all along because we can turn it on by these techniques. So you know, I think there are a lot of engrams which are not accessible. So we have a lot of memories that we can access. I know they're, you know, early on, or maybe even now, when they have brain surgery, sometimes the surgeon will probe the brain with a little electric shock. I'm not sure I'd want them to do that for me, but the person, it will trigger a memory. So it does, it's consistent anyway with specific regions of the brain being where that memory is stored. And so you can do that. So I suspect that there are memories that a person can't retrieve that are still there.
Experience.
Yeah, interesting question. Well, we know that you know, there are different kinds of memory, and what we've been talking about is called episodic memory, memory of an episode or event that happened. And then we have the semantic memory. And so if someone tells something to someone else, it is possible, I suppose, that they could incorporate that into their own memory. Sort of like the false memory we talked about with the mice, that engrams get linked together and create a memory which isn't real. So I could see how that might happen.